Plant Phenome Journal. And our Plant Phenome Journal webinar for today is Holly Lane. She's currently an inside sales consultant at the Meter Group. Previous to that, she was actually a master's student with myself. And the editor that handled her journal, Michael Gore, Dr. Michael Gore, who is at Cornell, is currently on faculty development leave or sabbatical or whatever you want to call it. So he was unable to host. So I'm going to host today, and we're going to talk about her really interesting work, although I'm completely biased here, on phenomic selection and prediction of maize grain yield from near-infrared spectroscopy of kernels, a project that really she did a lot and was instrumental for in designing some components of it that I hope she's going to talk about today, and roping in Dr. Jose Crossa from CIMMYT and some of his collaborators to really extend and expand the project. So without further ado, Holly Lane will be giving this month's webinar. Holly.
Thank you, and thank you for the invitation to speak. Okay. So yeah, as Seth said, I'm going to talk a little bit about sort of the project that was the focus of my thesis work when I was finishing up my master's at A&M. So just to kind of give a little bit of background in terms of plant breeding trends, since this was a plant breeding project. Plant breeding is, as many of you know, an ancient human endeavor, but we expect that it likely began around 10,000 years ago. Though, of course, it looks quite a bit different now than it did back then. But traditionally, we've sort of always had this focus on phenotypes, or what we can visually see. So picking the best plants based on what we could see with our eyes, or what we could taste, or something like that. Though more recently, there's sort of been this increased focus on the underlying genetics. And this has changed sort of the field quite rapidly. And so what we've sort of run into is this problem where we have increased our focus on genetics, but sort of our technologies for phenotyping have created this bottleneck. So field evaluations are time, labor, and cost intensive. And so we sort of want to have this goal of developing indirect, efficient, and more accurate methods of phenotyping to sort of release that bottleneck. So near-infrared reflectance spectroscopy has been used in our program pretty extensively before this, since it's reflective of chemical composition. So in our lab, we've built calibrations to use it to predict protein, starch, oil content of grain. It has quite a wide range of applications across many different industries because of its ability to detect chemical composition. And it has actually even been shown to successfully differentiate between species and even between genotypes within species. And so sort of this question that we're asking is, does NIRS have value not just as something we can extract phenotypes from, like protein content, starch content, and oil content, but does it have value sort of in and of itself as phenomic data? And so you can sort of think as of a phenome as the expression of a genome in an environment. So just a little more background on how near-infrared reflectance spectroscopy works and the science behind it. So as you move across different wavelengths, the spectra are going to fluoresce depending on what sort of chemical bonds are present. So the study that sort of sparked the concept for this project was something that came out in 2019 in G3, and this group, Rinson and al., coined this idea of phenomic selection, and they worked on wheat, which is an inbred crop, and poplar, which is a hybrid crop. And what they sort of did was they used near-infrared reflectance spectroscopy data in complementation and in replacement of genomic data. And so what they found was, in many cases, it was just as accurate to use near-infrared data as genomic data to predict, but in all cases, using phenomic selection was cheaper. So you could collect no genomic data, use only near-infrared data, which is extremely inexpensive to collect, and then achieve just as accurate results. And they ran some models, and even when phenomic selection was as inaccurate as it could be and genomic data was as cheap, you're currently using genomic data in a selection protocol. So phenomic selection, as you probably noticed, is sort of analogous to genomic selection. So just some background on the genetic material from this study. It was a genetic study grown as part of a genome-wide association study, consisted of 346 hybrid lines, all crossed to TX714. And we had irrigated or well-watered WW and non-irrigated or water stress WS treatments. And this was grown over 2 years. So in 2011, the region of Texas, College Station, experienced a pretty severe drought. And 2012 was a little bit more average with moderate temperatures and timely rainfall. I think in a normal year, we irrigate around 2 to 3 times. And I think I was told that in 2011, they had to irrigate, I think 10 or 11 times, which is pretty significant drought. So these water stress lines in 2011 were extremely stressed, and a lot of those plots were actually harvested by hand because there was so little corn to be harvested at the end of the season. And this is the work of 2 of my co-authors, Yvonne and Gerald. So just to kind of show you the difference in environments here we had between years. So this is the yield distribution of 2011 and the yield distribution of 2012. As you can see, 2011 had very low yields, whereas 2012 had quite a bit more of a normal distribution due to that more timely rainfall and moderate temperatures and less drought stress. Our data was collected on a Thermo Antris 2 Fourier transformed interferometer, and all that really means is that we get a really high spectral resolution. So it was collected in the range of 10,000 to 4,000 wave numbers. If you're more familiar with nanometers or wavelengths, that's 1,000 to 2,500. We had over 2,500 whole kernel samples, and what I mean by whole kernel is that these samples were not ground, so this was a non-destructive process to the kernels. And then we had corresponding yield on a plot basis. So for every plot, we had a yield sample as well as near-infrared being performed on them, and this resulted in over 8 million spectral data points, which, as you can imagine, is pretty hard to visualize, but I've tried to do that here on the next slide. So this is what the raw spectral data looks like. As you can see, it's kind of hard to visually interpret anything that's going on here, but you do have to treat this data. And so I used a first derivative Savitsi-Goulet filtering method. And as you can see, this is kind of normalized the brightness of the bands. And so now we can kind of see a couple key differences along the spectra where there are really differences. So what this does is it helps us to determine more meaningful differences between samples. Even though you can kind of visually see what's going on, you can't really visually interpret any meaning to this. And so traditionally, to develop a prediction method based on near infrared, you have to select some representative calibration samples, and then you send them for wet chemistry. And then you use those true values to train your model. And I'll explain that a little bit more in the next slide. So traditionally, when you build a near infrared calibration, you'll kind of scan all of your samples, and then you will plot them out based on your principal components and kind of see, make sure that you're sampling representatively from your data set. And so what you'll do in a traditional setting is you would select samples for both calibration and validation. And so all these colored dots here, you would send out for wet chemistry and get sort of what you're defining as a true value. And then you would build your model based on this calibration set, so these red dots here. And then to determine some level of accuracy that you're achieving, you would then test your model on the validation set. And so you kind of know what the true values are from your wet chemistry, and then you can see what your model is predicting. And that allows you to sort of estimate the level of error that you're getting with your model, and figure out if it's an acceptable level of error, or if you need to rebuild your model, maybe send some more samples in for wet chemistry. And then after you've calibrated and validated a model that you think is sufficient, you then use it to predict your unknown samples. So these open dots here, the white dots. However, in our case, we have our sort of wet chemistry, our true values for all of our samples, because we had yield for every plot. And so because of this, I just selected one single test set, since we did not have to send anything for wet chemistry, and essentially any sample could have served as a calibration or validation sample. And again, this is very similar to how you would develop a genomic selection model. You would select calibration validation samples, and then use that model to predict unknown samples. So this project used a couple of statistical methods. The first one we're calling our Phenomic prediction method, and this is our partial least squares regression. This is a statistical method that's very commonly used in developing near-infrared calibrations, and so that is why we selected this sort of as our baseline method. And with our Phenomic prediction method, we were predicting on a plot basis. And this, as I said, sort of mimics the traditional use of near-infrared data. And then we also had some phenomic selection models that were designed. And for this, I collaborated with Dr. Jose Crosa and his team. He's a biostatistician down at CIMMYT, and he deals with a lot of genomic selection models. And so I was able to get him on board, and he was able to develop a NIRS BLUP. BLUP here standing for Best Linear Unbiased Predictor. So to any geneticists or breeders in the call, that's going to be sort of working similarly to a GBLUP or a genomic BLUP. So what that's doing is it's looking at the NIR and trying to estimate kinship matrices. So trying to judge how related things are based on the similarities of their near infrared spectral bands in a way that normally is being done based on SNP data. And then his group has also developed a new multivariate method called functional regression. And that's sort of meant to deal with data whose information can be represented as curves or functions. And as you kind of saw before with the spectra having sort of that curved nature, this method, as you'll see in a bit, actually worked really well to handle this sort of high density functional data. And in contrast to the phenomic prediction, these methods were done on an entry basis. And so predictions were made for an entire hybrid line where yields and near infrared data were averaged sort of across each hybrid. And these sort of more so mimics genomic data approaches in contrast to the phenomic prediction, which was more of a traditional NIR use. So these are the results of our partial least squares regression model that was predicting within the experiment. So if a sample was held out for the training set, all iterations of that same hybrid or pedigree were also held out. If something was seen in the training set, it was not seen in the test set, and vice versa. And across all environments, we had a Pearson's correlation of 0.84. And I think it's important to note that this is actually a little bit better than even you might expect to have a calibration perform on predicting something like composition. And so we also wanted to check and see if was our near infrared sort of just corresponding to some sort of compositional aspect of the kernels. So we had protein, starch, oil predictions for each of these samples. And so what I did was run a simple linear model to see if there was a correlation between any of these compositional aspects and yield. And what you can see is there was quite a significant correlation with protein and yield. And even in combination, when you put protein, starch, and oil into one model. But The highest I could reach was 0.63, which has still not come close to the partial least squares regression model using all of the near-infrared data. And so while I think that the near-infrared is capturing some amount of composition and that's correlating to yield, there's really kind of some more to the story and something else that the NIRS is capturing that allows it to estimate how the parent plant performed. And so just to kind of compare our three statistical methods here in one table, this is a cross-validation scheme we called CV0. And this means that we are predicting one environment using the information from all of the other environments. So for example, here, we're predicting 2011 data using 2012 data and vice versa. Here, we're predicting water stressed 2011 using information from all three other environments. And so some major takeaways here to note are that it appears to be better to train your data on the stressed environment. So we could predict the well-watered and the 2012, which we know was less stressed than 2011, much better than the reverse. And that was true across all statistical method types. We also saw that in terms of the phenomic selection methods, functional regression tended to outperform the NIRS BLEP method. And I think that's really interesting because the functional regression is sort of a novel approach to handling this kind of data. And that original paper by Renson et al. Implemented a similar NIRS BLEP approach. So I would maybe argue that going forward, researchers who want to look more into this might be interested to see if functional regression handles their data as well as it handled ours. And then just a little bit more from the results of these phenomic selection models. This is a different cross-validation scheme we deemed CV2, and this mimics sparse testing. So what this means is that the model has seen all hybrids and all environments, but has not seen each hybrid in each environment. So for example, if the model has seen genotypes A, B, C, and D, and has seen environments 1, 2, 3, and 4, it might not have seen genotype D in environment 3, so it attempts to predict that based on the rest of the data it's been given. And as you can see, the functional regression here significantly outperformed the more traditional NIRS BLEP approach. So all of this was done on that genetics GWAS study that I mentioned earlier, but we wanted to test and see, was this calibration going to be something that could perform on breeding material and material that was unrelated to what the model had been trained on. And so here I have 6 breeding tests that were also grown in 2011. And this is that model, that partial least squares regression model that was trained on the genetics, the GWAS data. And this is its ability to predict on this breeding material as a second validation set. And as you can see, sometimes we're able to predict quite well, like with this test for AF. AF here standing for aflatoxin. These were aflatoxin resistance trials. Even if it was a location that was unknown to the model. So our original data is trained on College Station, but we were able even to predict a test in Corpus Christi with quite high accuracy, 0.68. However, sometimes we can't predict well at all, as you can see sort of here with some of the earlier aflatoxin tests. And the reason why this is happening is something we don't quite understand yet, but I think once we can kind of parse this out, that will allow us to be able to sort of optimize these protocols and will allow near-infrared data to become more valuable to breeders. So how can NIRS detect grain yield? And the short is we don't really know, but we kind of have some guesses. And so we think that some of it is that it has an ability to capture genetic relationships. But if that was the only thing that was at play, then the phenomic selection models would have greatly outperformed the phenomic prediction method. And that was not the case. It also could be that the models are detecting some composition in the kernel related with yield, which we sort of tested. But also if that was the case, we would have expected the plot-based phenomic prediction methods to greatly outperform the phenomic selection methods. And that was not the case either. And so what we think is it's likely some combination of these. And I think this also brings up some really interesting fundamental research questions for the future of near-infrared reflectance spectroscopy and sort of maybe its capacity to be used in ways that we didn't think possible before. Of course, as with any study, we had some limitations. The first one being that yield is a complex trait and it's affected by many factors. So it's kind of hard to pin down, all of the things that are influencing yield and yield capacity. But I would argue that NIRS offers sort of a potential to complement current selection methods. Also, the scans from this study are post-harvest. So I think that future studies should attempt to scan prior to harvest, and that might be more useful to be able to scan before you plant and then potentially predict how things will perform, and then maybe not planting things that you know are not going to perform well, and use it sort of as a negative selector. These models were assessed in known or pseudo-unknown environment. And what I mean by that is NIRS was collected in each environment. Unlike genetic data, which is just collected one time, this NIRS information was collected not just in every environment, but also in every plot and every environment that we looked at. But we do know that stressed environments appear to be better for training and building models. So perhaps in the future, when we try to figure out if we can just collect NIRS one time, making sure that the material is somewhat stressed to be able to really capture everything that the NIRS can tell us. Unknown pedigrees and known environments show promise. So if we know how the environment's performing, the model does a pretty good job of predicting a pedigree that it hasn't seen before. So overall, NIRS definitely seems to be a strong candidate for this sort of low-cost indirect phenotyping I talked about in the beginning, as plant breeding as a profession really needing right now. Ultimately, I think this could reduce the costs associated with field testing for breeders, and also, could maybe expand to crops that don't have a lot of genetic data available to them. And so therefore genomic selection isn't something that's feasible, even if they had the money to implement it. So in its current form, the potential uses include improving plot combine yield data. So if you were able to maybe mount a near-infrared scanner onto your combine, this would even save you a step of having to collect and then do your scans after harvest. You could do it sort of in conjunction with the harvest happening on your plot combine. There's potential that you could maybe harvest a single ear from each plot and just scan that with the near infrared, and then only harvest the plots that you think showed promise. And then, as I mentioned before, negative selection. So maybe it's not very reliable at telling you what the best lines are, but it is reliable at telling you what you know is not going to be good. So you can use it for that before you subject better lines to more expensive screenings. So with that, I'd like to acknowledge Dr. Seth Murray for being my advisor for this project, everyone in the lab that helped me with this, and of course my collaborators, Dr. Jose Crosa and his team at CIMMYT. And with that, I'll take any questions.
Okay, well, thank you, Holly. I do want to apologize for any disruptions that may have occurred. One of my students said that there was some video that I apparently couldn't see, but I followed all the Zoom procedures and locked down everything as best I could. So apologize for anybody who saw something they didn't want to see. But great talk, Holly. Of course, I think it's excellent work. What I've done is I've opened it up for questions in the chat box. I'm the only person who can see the chat now because, again, I think people have figured out a way to get around Zoom controls now. So I guess I'll start off with a question, Holly. So this was kind of a challenging project. I think most people don't try and work with collaborators online that they've never met before, but I guess you were well prepared for the current situation. So what did you find most challenging in this project as a student coming into it?
I guess hindsight is always 20/20, right? It's easy to kind of see now what skills you wish you would have had at the beginning. And I think maybe earlier on, I wish I would have maybe brought in someone who who was more familiar with the genomic selection process. And I think maybe then I could have emulated this project a little more in line with that earlier on. But I think overall it ended up being nice to sort of have a comparison to the more traditional way that we would approach using near infrared data, and then sort of having that second level comparison of a more genomics based approach.
Okay, great. We got a couple questions here from the audience. Dan Northrup is asking, would you expect this approach to work on other tissues besides grain?
Oh, that's a good question. So I know the Rincent paper did look at using parts of the plant that were not just the grain. So in wheat, I believe they used leaf tissue and grain. And their results, I believe, were that the grain was a little bit better. But they also had success in poplar. And for that, they looked at, I believe, the bark. So I think it might just sort of depend what kind of traits you're attempting to predict with your plant. And then maybe, selecting a plant part that might be representative of that.
Okay. And Marcio Resende asks, hi, Holly, great chat and great work. I agree with the assumption of, can you hear me?
Yes.
Okay. Hi, Holly, great chat and great work. I agree with the assumption of a combination of pedigree and composition leading to high accuracies. The question is, did you look at the accuracies in predicting composition in your CV0? Did the experiment where NIR BLUP have a higher accuracy show lower composition accuracy? So the first question, did you look at the accuracies in predicting composition in your CV0?
So we did not look at the accuracy of prediction composition. Seth, you might be able to answer a little bit more what kind of accuracy we expect with our lab's composition calibrations. I think that might answer that question a little bit better.
Yeah.
And then,
go ahead.
And then I don't know that I quite understand the second question.
Okay, so our accuracy in predicting protein is somewhere in the 90% range, with starch and oil, which were the other two that you used in our sort of simple NIR calibration. I think oil is like around 30% and starch is around 60%, but don't quote me on that. The second question was, did the experiment where NIR BLUP had a higher accuracy show lower composition accuracy? Yeah.
I don't know that was something that Rincent looked at. And I know it was not something that we looked at in this study.
Yeah, I think maybe there was a little bit of a miscommunication in how that occurred. I'm going to skip to the next question from Lee DeHaan. Did the NIR data make a relationship matrix that was well correlated with the genomic relationship matrix?
Yeah, so that's a great question. If there's anyone who is interested in taking in a deep dive into the genetic data from the study, that is publicly available. It is not something that we looked at, but I think it would be really interesting to have that direct comparison between a genomic selection model being performed on this data and then compare it to the phenomic selection.
Okay. Relin Alvarez asks, I'm not familiar with the literature, so sorry if this has already been done before. You're trying to predict something very complex, but is there any information on how well the method could predict traits that are more directly measured by NIRS, such as protein level?
Yeah, so NIRS is used very widely across many different industries to do that. And so we do that in our lab to predict protein. So it's got some very established protocols for that already. This study was mainly trying to see if we could sort of take that a step further.
Okay. Merrill asks, if you plan to use NIRS for pre-screening, can still stress be applied to seeds to improve the results? If so, which stress at which stage specifically will be better?
That's a good question. So I think maybe that's, should the stress be applied to the plants that you're going to harvest and scan, or should you apply stress after you scan and then are planting those seeds? And I think we don't know yet. You know, in this case, we definitely had significant drought stress and heat stress, and I think that worked well for us. But I think, you know, the details of these protocols are still yet to be discovered.
Okay. Marcio Resende has another question. One hypothesis is that in the water stress environment where NIR BLUP is higher, all you are using is relationship. In the other, where partial least squares is higher, it could be a combination of relationship plus composition.
Yes, yes, and I think that that's definitely probably true. And I didn't have time here to present the phenomic selection model's results on that validation breeding material, but the phenomic prediction did perform better on that sort of genetic material that was very different from what the model had been trained on. And I think because maybe that plot prediction, that plot-based prediction method with the partial least square is maybe relying a little bit more on composition than the phenomic selection methods. It sort of it was more robust to be able to withstand new genetic material because it could rely on composition instead.
Okay, I don't know how to say this. M.A. Numala, thanks, Holly, for your great presentation. My question is how the functional regression approach is different in other models. This model can be applicable to all the crops.
So the functional regression questions might be a little out of my wheelhouse because they were implemented and designed by Dr. Croson and the Montesinos Lopez brothers. I know they have a couple papers I'd be happy to share on sort of how it's working. But I think it is a new method and we had success with it here. And so I think that maybe it has a lot of opportunity to be used in more traditional settings.
Great. Scott Wild asks, for some, so yes, he can't send a chat to everybody. Again, I think we were, I guess we've made the big time because we were victims of Zoom bombing. So congratulations, Holly. His question is, do stressed environments improve your model because it includes an extra degree of freedom to the model?
I don't think any degrees of freedom are being added there. I think sort of you see this in general as a phenomenon in breeding, that a stressed environment sort of allows more separation between lines. You know, the perfect environment, everything sort of does pretty well. So if you put things in a stressed environment, you're really going to start to see a separation between things that do well and things that don't do well. And I think that was reflected in the NIRS data.
Okay, that's the last question I have in the list right now. Was there anything? Feel like there was one that we didn't quite understand, but I haven't seen any more from the person who asked that. So I guess I'll ask the last question, unless we got another one coming up. Here we go. Relin Alvarez, related to the last question, maybe it would be beneficial to apply different types of stresses in your training experiment.
Yes, I agree. I think now that we've done this, we sort of know how to design a much better experiment for this in the future.
Yeah, and I think, you know, just to reemphasize what Rincent found and what you found was that the stressed environment was a better predictor. But we only had a couple environments in this study, and I think if you looked at deploying this to an industry program, for instance, where they might be collecting 100 environments over many years and saving all that data, I think you would be much more likely to hit some optimal stress environments than you would be, you know, just with our sort of, you know, follow-up study to the original Rincent study. We used the data, or Holly used the data that she had. So yeah, probably apply more stresses in the future and targeted stresses. And the Genomes to Fields project would be great for that.
Yes.
I guess last question. So with the Rincent paper, you know, I kind of felt like it was a really big deal. I know Jose Crosa with some collaborators in Jesse Poland's group and Mike Gore's group have been working sort of around the edges of phenomic selection. What do you think the biggest barrier is for people to adopt phenomic selection as a new technique?
I think maybe because it's such a different way for this data to be used than it has been used in the past. And you know, the idea that it could contain better information than the genetics data maybe doesn't make sense intuitively at first. And I think, you know, another barrier would be if a program doesn't already have that piece of equipment, they can be a little bit expensive. And so sort of to have to buy new equipment and to develop some new data collection protocols, I think is sort of daunting.
Great. Well, if we don't have any other questions, I want to thank Holly for a great presentation. I want to thank her for deciding to put her article in the Plant Phenome Journal. This is exactly the type of